It's an international group. There's a few of us in there. There's Dr. Leroy, that is the founder and the maker of Biotech's Exosome Kit, the one that Dr. Janira is actually currently using and teaching with. And then there's Dr. Satwinder in group. He is anti-aging doctor, and he's actually very, very advanced in his bioidentical hormone replacement therapy for in Malaysia. Okay. And then there's also Dr. Prabhu Mishra. He's practically stem cell research and doctor for in the world platform and globally. He's also will be answering question there. So if in case you guys have any question, you can actually bring it up in the group anytime, and we are there to actually help you understand how to actually use regenerative tools. Okay. So without further ado, let's go back into Autologus Exosome. Okay. So what I'm trying to say tadi ialah, what we want everybody to know is Autologous Exosome and exosome itu tidak sama. Autologous Exosome ialah resipiennya dan donornya ialah orang sama. Whereas exosome tu seringnya donornya ialah orang lain and then resipiennya ialah orang lain. Kenapa? Okay, so usually Autologous Exosome ialah exosome yang anda buat sendiri di klinik anda. Tapi kalau exosome itu yang popularly known yang boleh membuat pelbagai proses regenerative, that one is actually known as mesenchymal exosome, and that is usually made in the lab. Mereka selalu bikin itu di lab, and then di sana mereka bisa quantify it. Mereka boleh masuk dalam botol, and then and then it's being known as, okay, it carries maybe 300 million cells, 480 million cells gitu. And then you beli dalam size 15 ml, kadang-kadang 3 ml, 8 ml. Tapi kalau autologous exosome itu ialah exosome yang anda bikin sendiri di lab anda sendiri. Dan macam mana kita nak bikin, how can we do those autologous exosome, nanti Dr. Janira akan jelaskan. Okay, so pentingnya kita faham dulu exosome. Exosome ialah, kalau dilihat, ialah extracellular vesicle. Saiz dia selalunya di antara 30 sampai 150 meter, 150 nanometer. Okay, dan exosome ini kita dapat daripada secretion of all cells. Okay, so yang penting ialah kita harus tahu saiznya ialah antara 30 sampai 150 nanometer. Kenapa? Kerana that's the only way when we actually send for testing to the presence of this size particle, that means that is extracellular vesicle of autologous exosome or exosome. Sini exosome as general. Okay. So seringnya exosome ini dia carry ada protein, ada lipid, ada growth factors, ada cytokines, okay, ada DNA fragments, selalunya ialah messenger RNA and microRNAs, okay. So fungsi dia selalunya ialah natural biological messenger, okay, is for cell-to-cell communication. Dia regulate gene expression, modulate immune responses, and dia membantu tissue repair, okay. So atas exosome ni, di surface exosome ni ada ligands, ada and markers, selalunya tetraspenin CD9, CD63, CD81. Okay, two ways untuk tahu exosome. Satu ialah melalui size dia, dan untuk mengetahui exosome ini ialah exosome, kita buat test on CD9, CD63, CD81. Only then you know that this is actually exosome. Okay, so apa beza dia dengan stem cell? Okay, paling penting ialah active signaling package of stem cells. Kalau menggunakan exosome, kita tidak ada risk untuk tumor regenesis atau cell rejection atau uncontrolled proliferation. Kenapa? Kerana stem cell itu ialah pluripotent cell. Benar? Okay. So, tapi kalau exosome, remember this, exosome is not a cell. Kita tidak bisa mengatakan exosome itu satu sel. Exosome itu sebenarnya ialah signaling factor saja, ya, sejenis particles. So, exosome tidak mempunyai sel. Exosome itu adalah a simple signaling factors. Kerjanya ialah biological messengers, adalah for cell-to-cell communication. So, kalau kita ketahui dulu ya, okay, bila exosome was actually introduced, ramai yang fikir exosome tu ialah just a carrier untuk dilupuskan. Maknanya it is just a tong sampah. Okay. Tapi tidak. Sekarang kita dapat mengetahui kalau if you see into the cells, okay, into dalam sel, dalam cytoplasm, dalam sel, bagaimana exosome itu dibuat, or how the process of exosome is actually happening. Nanti Dr. Jana akan show. Okay, dan kita akan faham exosome ni dia dua jenis. Satu ialah untuk ke lysosome untuk dipecahkan dan untuk dibuang. Satu lagi dia keluar sel jadi signaling factors. Okay, so again, Dr. Jana hanya nak menunjukkan advantage antara exosome dengan stem cell. Kalau dilihat di sini ya, okay, this is stem cell yang besar ini. Okay, dan exosome ialah yang kecil-kecil ini. Okay, this is the, so stem cell itu ialah 180 kali lebih besar daripada exosome actually. Okay, and the next slide. Okay, so how the formation of exosome comes in. Okay, so biogenesisnya simple saja. Okay, so kalau dilihat gambar ini terlalu kecil ya. Okay, okay, so kalau dilihat setiap sel yang ada bilayer membrane, okay, bilayer dual membrane, okay, bilayer lipid membrane, okay, dia pasti akan ada sel di dalamnya, I mean dikenali sebagai sel, okay, dan di dalamnya akan ada nucleus, ada mitochondria, ada lysosome, ada Golgi apparatus and everything, right? Okay, so apa yang sebenarnya berlaku ialah sel kita ni pintar. Dia ingin mengetahui apa yang ada di cytoplasmnya. Dan seringnya it is a continuous process. Dia akan selalu melakukan sampling of the cytoplasm. Bagaimana dia sample cytoplasm? Very simple. Dia ada cara ialah pinocytosis, phagocytosis, pinocytosis, dan satu lagi ialah RME mediated. Okay. Kalau pinocytosis ini, dia akan minum apa yang ada di luar. Kalau phagocytosis itu ialah how macrophage works. Okay, and RME mediated ni ialah satu yang actually diminta oleh, sorry ya. Okay, phagocytosis ialah, I mean the last RME ialah apabila cell have a signal di dalam cell itself, dalam sel itu untuk bina untuk build signaling factors. Okay, so kalau dilihat kemarin Dr. Jana boleh lukis, hari ni nggak bisa lukis, so just bayangkan sajalah. So ini ialah sel, okay, so ini ialah cytoplasm, ini ialah sel, okay, dan apa yang berlaku ialah sel membran ni akan fold inward sebegini, okay, dan seterusnya sel membran ni akan fuse. So apa yang dia dari luarnya masuk and fuse. Okay, so yang dalam ini sekarang will be known as early endosome. Okay, dalam early endosome ini pelbagai jenis faktor akan masuk. Okay, selalunya all kind of factor will be signaling factors. They can be mRNA, it can be protein, okay, whatever, dia akan masuk ke dalam itu. Okay, and then dia akan jadi daripada early endosome, dia akan non-stop, dia akan tambah. So cell fold in, membrane fold close, dan dalam ni akan form vesicle. So dia akan jadi beribu-ribu, berpuluh-puluh atau I don't know how many, tapi too many vesicles, okay. Dan tergantung sel itulah ya, okay. So sel ini dia akan fuse, okay, dan dia akan menjadi late endosome, okay. So kalau sel itu contain information atau mempunyai information yang tidak dibutuhkan oleh badan, dia akan hantar ke lysosome, dan di lysosome dia akan pecah. So that's how our body pilih apa yang harus dibikin atau tidak dibikin. Okay, tapi kalau codingnya cukup, kalau informationnya cukup dan dia ingin hantar signal, okay, usually this is determined by our DNA lah ya. Okay, so dia akan keluar, dia akan fuse dengan plasma membran. Okay, so sama, nak terbalikkan, dia akan buka lagi ini, dan di bawahnya dia akan keluar, dia akan jadi satu vesicle, dan dia akan mulai signal dia. So exosome itu dia bisa signal locally atau dia bisa jalan jauh ke mana-mana saja. So far everybody okay? Okay. So I think that is already a very detailed explanation, yeah. Okay. So this is the structure, okay. tadi Jana bilang, okay, it can has pelbagai jenis structure dalamnya, okay. And then, okay, so other things cell secrete selain daripada exosome, apa saja sel secrete? Okay, this is general ya, bukan exosome saja. So tadi kan Dr. Janira bilang only cell akan secrete something kan. So one of the thing that is secrete is actually exosome. Tapi kalau tak secrete, dia akan masuk ke dalam lysosome. So if you can see, this is all the endocytosis. Nampak tak dia endocytosis, this one. Okay, then dia akan fuse and then dia akan jadi early vesicle body, then turn into late. Kalau yang this is unfavorable, dia akan masuk lysosome, will be destroyed. Kalau favorable, dia akan fuse dengan membran dan dia akan keluar jadi exosome. So, this is how exosome is actually formed into your body. Okay. So, selain daripada exosome, badan kita juga mengeluarkan apoptotic bodies, okay, cytokine growth factors, lipoprotein particles and other nanoparticles. Okay. Microvessel juga dikenali ectosome, itu dari dalam luar sel. Okay. And next kami sedang belajar ialah nanoparticles, ialah exomeres, yang nombor 5 ini, okay. Recently discovered, okay, non-membrane bound nanoparticle, distinct, dia sangat beza dengan exosome. Tapi this is what so far everybody been seeing that maybe carrying DNA factors. Kemungkinan, tapi nggak pasti lagi. Okay, so kita lagi belajar. Okay, so tadi this is the whole structure of exosome. Again, Dr. Janira akan bilang, it's not a cell, okay? It's just a signaling factors, okay? Dia bukan satu sel, okay? So dalamnya ada apa? Tadi Dr. Janira ada bilang, okay? So ligand ni yang menentukan dia akan dock ke mana. Maknanya dia akan sel mana, reseptor mana, ligand ni akan ligand yang luar ini CD81, CD9 dan CD63 ni akan determine dia akan ke mana. Tapi at the same time, dia ini juga yang kita guna untuk mengenal pasti ini adalah exosome, okay? So dalamnya ada nucleic acid kan, ada amino acid, ada metabolites yang Dr. Janira bilang, okay? So kerjanya apa sebenarnya? So dia akan ke signal, dia akan dock ke sel lain dan dia akan mulai signalnya. Signalnya hanya signaling mainly untuk regulation of gene transcription and translation. Selalunya signalnya harus yang baik, survival and proliferation, reproduction and development, angiogenesis and wound healing, waste management. Okay, tadi waste management di kalau ke lysosome. Host microbiome interaction and viral immunity. Okay, balance of immune response, okay, and regulation of central and peripheral immunity, okay, receptor ligand signaling, dia bisa juga pergi untuk bilang pada cancer cell, this is for you to die. Apoptosis is a signal, is a messenger. For cellular differentiation and neoplasma, cellular migration and metastatic disease, and metabolic reprogramming and regulation. Okay, so kalau dilihat ya, dia akan dock to recipient target via surface ligands, dia akan fuse dengan membran and dia akan deliver cargo. Cargo apa? Cargo yang ini, nucleic acid, jarang DNA means mostly RNA. Tapi yes, exosome boleh carry DNA. So this is where the question comes in regenerative medicine hari ini. Do you want to carry somebody else's DNA in your body? So that's my question when you actually use stem cell or exosome. So that's why the best is always using your own exosome, meaning autologous exosome, donor is you, recipient is you. Tapi kalau you are using somebody else exosome, walaupun dibuat di lab, okay, has a lot of positive signaling, tapi kemungkinan you're also being exposed to other people DNA and other people RNA. Bukan your generation, but the next generation, they might show different response. So hence today, kalau me, I will always choose to use autologous exosome. That is your own. Make sure it's your own donor. Recipient is also you. Okay, so what is amazing about exosome ni ialah dia mempunyai bilipid membran layer yang sama dengan blood-brain barrier. So it can actually cross the blood-brain barrier very, very easily. Okay, so what do you call that? They can signal and they can actually penetrate the blood-brain barrier. So again, like I said, kalau you use stem cell or because stem cell, when you use the whole stem cell juga akan mengeluarkan exosome-nya sendiri. So when you are actually buying stem cell dari luar, the stem cell medium has to be grown in an exosome medium. So di situ sudah ada exosome. So exosome ini yang carry other people DNA can also cross your blood-brain barrier. Okay? Okay, so what is the main function of exosome here? Okay, tadi ada beritahu. Okay, exosomes are not waste. Previously, they think it was a waste, tapi they are actually a biological messenger. Okay, cell actually menggunakan exosome ini untuk talk to each other to a distance beyond direct to direct cell contact. Okay, and then tadi dah I already tell you what they carry: micros RNA, reprogram gene expression, yeah, reprogram gene expression in recipient cell. That means daripada your own one cell to another cell. Okay. Protein and enzyme modulate immune response, tissue repair, lipid, cell signaling, and membrane fusion. Okay. So maintaining cellular balance. Okay. So cells also use exosome to export unwanted material. Contohnya, dia boleh lock this thing untuk send toxic protein, damage DNA, or damage RNA or excess metabolite. Okay, so contohnya, if neuron release exosome to clear misfolded protein, cancer cells use them to dump stress molecules. So di cancer cell juga ada exosome. So this is where it gets very, very dangerous. Okay. Role in health ialah immune system, okay, regeneration, Dr. Janira dah bagi tadi, brain function, okay. Kalau role in disease, okay, cancer cell can hijack exosome, okay. And bila cancer cell hijack exosome, dia boleh guna untuk promote angiogenesis and immune evasion. So this is the first key, guys. You're not supposed to use exosome on cancer patient, especially active or acute cancer patient, okay? Unless you have already ensured that the patient is totally under regression, okay? How you want to do that? We have a masterclass coming on the 23rd and 24th to actually tell you how to choose a proper candidate. Okay, once we have everything drafted out, Dr. Jenna will also share, okay? Tapi again, just remember, viruses also guna exosomal pathways to spread viral RNA protein. So virus boleh hijack your exosome, okay? And then, use it to spread their RNAs, okay? So bila your candidate or your recipient is very high in chronic inflammation, especially during active infection. So again, that means I'm trying to say here, patients who are having infection, no exosome. Please don't use any kind of exosome, okay? So we can also use for face exosome. So example, if you have active acne, okay, you're not supposed to inject exosome there because that exosome can be hijacked and you can create more damage there instead of regeneration, okay, or repair, okay? And then kalau daripada evolutionary perspective, okay, this is our survival mechanism. So instead of taking signal randomly, okay, cells masukkan dalam package information in a stable lipid vesicles. Okay, again, vesicles here is not a cell that can survive in any system. That means any pathway dalam blood, limb, and bisa travel long distance. So that's our body way of survival, okay? And this is to ensure precision in communication and dia bisa adapt in stress condition. Okay, so now comes to the point of autologous exosome and allogenic exosome. I think dari tadi Dr. Janira already explained what's the difference between autologous and what's the difference between allogenic. Okay, so autologous is derived from patient's own cells or blood. Okay, bisa seperti isolated from PRP, serum atau bone marrow aspirate. Okay, this is very personalized, very customized. Okay, immune tidak akan ada masalah kerana ia akan compatible. Okay, it will reflect the patient current metabolic and genetic state. Again, I'm going to say this. It will reflect patient current metabolic and genetic state. Ini sangat penting loh. Kalau contohnya badan anda homocysteine-nya sangat tinggi, D-dimer-nya tinggi, and ESR-nya tinggi, okay? And then you give exosome pada patient ni, especially autologous exosome, yeah? The result is not going to be good, guys. Okay? So don't do it. So the patient before a therapy of autologous exosome is crucial and it is mandatory. Dr. Janira again saying it's mandatory. Okay, I think Dr. Vidya udah faham. Okay, she'll be taking lead of autologous exosome explanation in Indonesia. Okay, tapi this is what I would always say to everyone because hari ini there's many doctors who thinks exosome is only for regenerative, it's very good for anti-inflammation, it's very good for repair, rejuvenation. Tapi no. I already given you information tadi virus boleh hijack and especially main thing ialah it will reflect the current patient metabolic and genetic state. Okay, so kalau patient itu is very inflamed, if the patient is very oxidative stress, you bagi exosome, dia akan tambah lagi stress, especially autologous exosome, ya. Okay, so exosome may be less potent in age or inflamed patient unless they have to be preconditioned. Okay, now go back to allogenic exosome. So allogenic exosome ini ialah exosome yang is grown in mesenchymal stem cells. Okay, that means this is donor exosomes. Okay, so again, like I told you, stem cell must be grown in exosomes. Okay, and then that stem cells will also produce signaling factors. Okay, so it can come from umbilical cord, can come from placenta, or can be come from cultured bone marrow. Okay, so and they can be standardized. Mereka bisa dipilih, bisa dibikin lagi besar, scalable, and it can be available on the shelf. Okay, so it used in many clinical trial because it's large, consistent batches can be manufactured. Okay, so selalunya it is from young and healthy donor mesenchymal stem cells. Okay, so cargo ini memang akan penuh dengan growth factors, anti-inflammatory, microRNAs, and regenerative signals, because like I said, this is actually from umbilical cord and placenta, right? Okay, so more universal and pro-regenerative. Tapi again, kita harus fikir today in our umbilical cord and placenta, something called microplastic has also already invaded, not just microplastic, nanoparticles, and including what do you call that, forever chemicals is already there at your umbilical cord and placenta, and this presence of this unknown toxin can actually cause inflammaging atau destruction of lysosome. So again, you know, like I said, it's more safer to be choosing autologous exosome rather than allogenic exosome. Okay. Okay, my next steps. Okay, so this is just a basic pathway, ya. So kalau ramai yang di sini ialah integrative doctors atau doctors yang functional, okay, anda akan faham what am I trying to say here. Okay, so again, I'm saying autologous exosome, ya, not exosome. Okay, this is an exosome that you can actually grow in your own clinic. Again, Dr. Vidya will be taking the lead to actually show you how to do this. Dr. Leroy and Dr. Janira will come to Indonesia one day, and we can actually show you how to make your own exosome in your clinic. Okay? Okay. So steps and checklist is very basic. Please do baseline labs. Okay, please check for inflammation, oxidative stress, and make sure hormonals is actually in balance. Okay, selalu harus buat detox pathway. Okay, open the gut. Okay, detox the liver. Make sure kidney function is good. Okay, bisa melalui lymph node, bisa melalui sweat. Okay, clear your parasite, clear your candida, and of course your vaccine. Okay. Give the patient very high antioxidant and mitochondrial support seperti vitamin C, NAC, glutathione, coenzyme Q10, magnesium, and carnitine. That is basic. Okay. Akhirnya, make sure hormone juga is balanced dengan BHRT. Contohnya yang paling penting ialah thyroid hormone and cortisol. Kerana that's your stress hormone that will determine the inflammation in your body. Okay. Sex hormone juga harus look into, because that's mainly will correlate back into your growth hormone. Okay, retest labs and see clinical improvement. So when I say clinical improvement here is, it can be many types of clinical improvement. So example, kalau patient go through example, kalau a patient come and tell you that, oh, I'm having a cognitive decline, and after everything that you have done, a patient is telling to you right now, doctor, I'm having a better memory and everything is more clear to me. I'm no longer, blur. Okay, that is already a improvement in symptom. Okay, so those patient, that means the signaling in the body udah mulai signal yang baik. So you want to boost the signal, then this is the time. This is the best time for you to use autologous exosome. Okay. Okay, so these are the few baseline labs. Okay, this is how you want to know whether the terrain, meaning the body, is safe. Okay, so inflammation and coagulation, that's my target. Usually I tell to all my doctors to make sure that HSCRP target is below 1, ESR target below 10, tapi below 5 the best lah. Okay, if you're doing interleukin 6 and tumor necrosing factor alpha, kalau available, make sure that it's not there. Okay, the D dimer and fibrinogen, you must always check, because you know why? At the end of the day, we are actually drawing patient blood, and then we're going to centrifuge it. Before centrifugation, we are going to actually, incubate it, okay, to release all the factors, the signal, and then we're going to centrifuge it. If the patient D dimer and fibrinogen is very, very high, then the product that you yield is going to be very low. Okay, not saying you cannot do it, you can do it, tapi the product that you yield is going to be low. The autologous exosome that you're actually going to inject back into your patient is never dangerous. It can never cause any inflammation. It can never cause any immune problem, and it can never cause any clotting as well. That part all is actually very, very carefully being taken care of. So, but will you yield a good product? That is another question. Hence, make sure your patient D dimer, fibrinogen, everything is already stable. Okay. D-dimer, usually, currently, we see a lot of patients having very high D-dimer because of the vaccine effect. You see D-dimer, you see homocysteine, you see your inflammation marker. You can already know that this patient is clotting. Okay, so there are many other precondition that you can do for this patient. You can use nanocurcumin, nattokinase, you can use glutathione. There are many factors, or NEC or something like that. It will definitely help this patient. Okay. Otherwise, you also have to see if you have more advanced lab, then that's the second, detox capacity and oxidative stress, that you can actually look for. One of it is homocysteine, which is actually easy, below 9. Below 4 is not optimum actually. It's actually, there is something wrong with the methylation in the body. So you'll have to give more amino acid, because you want to target a better building, right?
Yang kita lihat dulu, kan bisa juga kita kontak orang BKKB nih.
Dokter Ana, please.
It's okay. Okay. Yang paling important at the end ialah metabolic and hormonal balance. Okay, Janira put this as number 3. Kenapa? Kerana this is the main thing that we have to see. This is actually the basic thing that we have to manage in our patient. Why? Because kalau HbA1c-nya tinggi, fasting insulin and glucose-nya tinggi, pasti we know that this patient is very highly inflamed, right? So make sure that this is under control. Okay, as I said earlier, cortisol AM and PM. Okay, DHEA is a very crucial. Thyroid, because it works together very closely with your mitochondria. Okay, sex hormone tadi, estrogen, progesterone, testosterone, because this can cause inflammation in their body. Okay, and main thing ialah make sure vitamin D, B12, magnesium, and zinc, and selenium, everything is okay. And RBC, what I meant to say is G6PD is normal. Okay, because you want to do preconditioning, you have to know that the RBC pathway is not disrupted. Okay, how do you want to do preconditioning? I think a lot of functional doctors, they know here today that, kalau for preconditioning, you cannot be giving high dose of vitamin C if the patient G6PD is not, G6PD is deficient, yeah, because otherwise then you can cause RBC hemolysis, right? Okay, so again, that's a step checklist. Okay, so this is step 2, detox and terrain reset. This is usually around 2 to 4 weeks. So this is your preparation towards exosome, yeah. Okay, so for me, like I always it's non-negotiable, yeah. So because toxic cells too definitely will translate into toxic exosome. So this is non-negotiable. So make sure you know how to do a good parasite detox. Okay, so because parasite will disrupt the gut barrier. Okay, it will hold on to heavy metals. It will create biofilm. Okay, so if you don't do this, if you don't do parasite detox, definitely your later detox will be less effective. And again, you cannot be absorbing nutrition through your dysbiosis gut or leaky gut. It's not going to happen. So hence, parasite detox is the first crucial part. Okay. Candida detox, this is also very important. After parasite, please put your patient under a candida detox, okay? Because Yeast overgrowth always fuel inflammation. I think we all know that will fuel our glucose, okay, will fuel our inflammation, okay. So if Candida still persists in your body, exosome will carry pro-inflammatory and neurotoxic cargo. I know nobody told you this before. Everybody will say that, you know, exosome is so good, exosomes for regenerative. Yeah, immediately they will have a regenerative effect, but this is in the longer run, guys. So that's why you'll have to make sure. Hence, that's why the topic today is what you should know before you inject. Okay, so be a responsible doctor. Okay, vaccine detox and post-vaccine protocol. Okay, this is my third tier in my detox tier lah, yeah. Okay, and then of course, drainage and clearance first. Okay, you give liver support. Example, you can use nutraceuticals. Again, Dr. Janira is going to be very, very clear here, yeah. If you want to go into regenerative medicine, so you can be an integrative functional practitioner, you can be a functional practitioner, you can be a homeopath, and then you can come into regenerative. These three factors is what makes a regeneration comes in. If you don't focus on understanding how synergistically your body is going to work together, you can never achieve regeneration. Okay, because you're talking about regenerative medicine is, you know, anti-aging, you know, reversing and everything on. And if you want this to be there and you want it to be maintained, then you have to always understand this. Putting in substances, example, synthetic nutraceuticals, that is actually very, very dangerous. So when you want to achieve regeneration, always choose natural. You'll always choose organic. And like for me, I will only use nutraceuticals. I will never use other than that. That is that's no question, yeah. Okay. So look for a smaller company, don't buy bigger company. I know no brand is very, very interesting, but when it comes to when personalized and customized, branding is not important at all. Okay, you need to find company that is reliable, that is trustable, because you want your patient to achieve maximum regeneration. So if you're using a synthetic polymer or synthetic made chemical or synthetic made product as your supplements, then you will never achieve any terrain reset. That is what I'm going to tell you very, very clearly. Okay, so your dandelion, your milk thistle, please make sure that they come from a trusted source. Okay, that's very, very crucial. Okay, coffee enema is one of the best way to actually do your bile clearance, but unfortunately today in Malaysia, every coffee that I see is covered with margarine. Okay, every coffee that is grown out there, it is adulterated with metal toxicity and manufactured by chemicals. Okay, roasted with chemicals. So to look for an organic coffee is very, very difficult. So make sure that you find a good coffee enema. Okay. Bowel clearance, example, using probiotics. Again, I would say, guys, probiotics is a reflection of your culture. So buying probiotics from America, buying probiotics from Europe is not going to help you because they are not made to digest your food. So always find food that is actually local. Okay, probiotics that is actually local. Look within Indonesia. Support local brand because these are the things that is going to break and digest your food again and maintain your gut health. Again, you can add on if you understand probiotics. If you need certain types of probiotic, example, if you have an infection and you need this, then yes, you can add. But that is single out probiotics. But if in case general probiotics, again, I'm going to tell you all, please choose local probiotics, not brand. Okay. So you can also use binders, MCP, charcoal, and chlorella. Okay, this is actually to catch all the metal toxicity at the same time to catch your microplastic from your gut. Okay, do a proper colon cleanse, do a proper digestive cleanse. Okay, give your kidney a support. Make sure your patient has a lot of hydration, electrolytes, and again nutraceuticals. Okay, put patient on epigenetic changes, example like, you know, far infrared light sauna, ozone therapy is amazing, dry needling, lymphatic massage. Also make patient to stay very zen with itself and the stress level is actually low. Okay. Again, chelation therapy is actually very, very important. This time today I'm here in Goa. Okay. And more others. Okay. So why detox first? Again, I've already mentioned earlier, okay, the molecular cargo package into exosome contains protein, RNA, DNA fragments, lipid, mirrors, and biological state of the cells that produce them. If the terrain is toxic, that means if your body is toxic, they will definitely give you back a toxic exosome. Okay, so if patient has high level of heavy metals, pesticide, persistent organic pollutants, or inflammatory cytokines, okay, those are the stress signals are selectively loaded into the exosome. Instead of promoting repair, these vesicles spread pro-inflammatory and pro-disease messages to other cells. Do you want this? Definitely no. Definitely not for your patient. Okay. So detox resets the terrain. Okay. It will open the detox pathway. Okay. And this is how we actually reduce the toxic noise in the cellular environment. Okay. Clean terrain is equivalent to regenerative exosome. Again, friends, clean terrain is equivalent to regenerative exosome. Okay, once the cellular environment is cleared and antioxidant reserve is restored, okay, exosome will be filled or will be enriched in growth factor, anti-inflammatory micros RNAs, and mitochondrial support signals. Okay, so these are the one that acts as true regenerative messenger. Okay, so in short, guys, detox is not optional. Okay, it is a filter that turns on exosome or a signal or a switch from toxic courier into a therapeutic tool. Again, it's not a cell, yeah, so it cannot think, it's only a signal. Okay, so this is my step 3. Usually what I do is I will also give the patient antioxidant and mitochondrial replenishment. I've not written NAD here is because in some country you can get NAD, some country you cannot get NAD. This is a very basic. So I think Dr. Vidya has more information on antioxidant and mitochondrial replenishment in Indonesia that she can actually share with you guys. Okay, but I've spoke to her the other day. I also understand that this is what she also do crucially for any of her patients. Okay, whether it's for exosome or not. Okay, so she has few protocols that you can actually get from her. Okay, so IV vitamin C, again, not G6PD deficiency ya kawan-kawan. Please make sure you check G6PD ya. Okay, because RBC will never be able to survive if G6PD is deficient, yeah. Okay, so IV vitamin C 5 to 10 gram. This is 1 to 2 times in a week. Okay, you can give NAC 1 gram. Okay, or you can give oral, or you can also give IV. Glutathione is amazing. Okay, but again, if you are giving for a cancer patient that is under remission, then you want to be very careful. You have to consider. Okay, understanding is the key. Coenzyme Q10 or ubiquinol, okay, that is actually very, very good because again, if in case you're having, lipid dysregulation, majority of us has, okay, this will actually help and will keep the inflammation from that area actually low, okay. Magnesium, I think I cannot say the importance anymore. It's very, very crucial, okay. Carnitine is actually very good to promote for your heart health and everything. Okay, so polyphenols that I will definitely give my patient 1 month or 2 months before I actually introduce exosome into their body if they are a candidate would be curcumin. I never discount. I use nano curcumin from Dr. Leroy because it dissolves in the water very beautifully. I've tried and I have actually reduced HRCRP and CRP in within 3 weeks. A significant amount of reduction by using Biotech's nano curcumin was amazing. Okay, resveratrol is another amazing ingredient, and EGCG is another amazing ingredient for your mitochondria. Okay. Okay, so this is not my forte. Hormonal optimization usually is what Dr. Satwinder does for all my patients. Okay. What I realized is over the years when I was actually treating patient, actually I don't treat so much. I always guide the patient. Okay, and when I give them nutraceutical, somehow their hormone markers will actually change because when I detox the liver, I detox the gut, and when all the precursor vitamins, example like iodide or zinc picolinate or selenium or iodide, I mean for thyroid function, it actually automatically improvises the thyroid function before I go to hormone optimization. So that's why I always use hormone optimization the last. So I detox first, second I replenish, and then after that I support, and then I retest the blood, guys. I retest the blood, and then only I decide whether I want to give hormonal or not. Okay, so please make sure that you are well versed on how to use hormones. Okay, because all of this only then you will be able to open regeneration in a patient body. Okay, so why it is important? Okay, dysregulated cortisol, usually chronic stress, adrenal fatigue. Okay, this will increase the reactive oxygen species. Okay, it can deplete the glutathione, weakens mitochondrial ATP production. Okay, so cells under cortisol dysregulation release stress-loaded exosome, yeah, carrying pro-inflammatory microRNAs and signal that perpetuate catabolism. So, restoring a healthy cortisol, like for me, I would use ashwagandha, something like that, okay, and melatonin for the patient to have good sleeps, to help them modulate their stress sleep hygiene, okay. Is one of the best things that I've ever done for my younger patients, okay. Adrenal reserve, your DHEAs, okay, you have to use to counterbalance your cortisol because they are very anti-inflammatory and they protect your mitochondria, okay. Again, low DHEA, they akan impact mitochondria biogenesis, okay? They can reduce the resilience to oxidative stress. Exosome from low DHEA, okay, often carry alarm signals instead of repair signal. Again, I'm telling you, exosome from low DHEA individual carry alarm system instead of repair signals, okay? So supplementing with a proper DHEA, again, what Dr. Janira is again saying is, make sure it's BHRT, bioidentical hormonal replacement therapy, not any synthetic hormone. Okay, so they can shift signaling towards repair, angiogenesis, and anti-inflammatory messaging. Thyroid hormone is very, very important. T3, okay, is the master regulator of mitochondrial respiration. Tanpa T3 yang cukup, mitochondria akan underperform. Dia akan menyebabkan less ATP and more oxidative leakage. For mitochondria function, exosome with weaker repair cargo. Remember that. Okay, impact RNA packaging, less angiogenic potential. Okay, so optimizing thyroid, sometimes dengan BHRT, again, like I told you, iodine, selenium, thyroxine, your gut repair, zinc carnosine, akan memastikan exosome are released from energy-rich, metabolically competent cells. Okay, sex hormones, estrogen upregulates mitochondrial biogenesis, protects neuronal mitochondria, increases antioxidant enzyme expression. Progesterone supports myelin repair for neuroprotection, stabilize mitochondrial membranes. Testosterone stimulate muscle mitochondrial activities, enhance nitric oxide. Very, very important for your hypertension, for your endothelial health. Okay, when these decline, especially dalam patient with menopause and andropause, mitochondria becomes very sluggish and exosome will carry weaker regenerative instruction. So use BHRT, restore the hormonal fuel that make mitochondria effective, efficient. Exosome akan jadi lebih regenerative messenger instead of neutral or inflammatory ones. Okay, so my final step is here. Repeat the CRP, oxidative stress marker, and homocysteine. Okay, clinical check a symptom. You can ask patient about their sleep, whether their fatigue is reduced. Improved skin tone is another thing that you can see for symptom improvement or the condition improvement. Sharper cognition. Majority of my patient, if we do step 1 to step 5 properly without even using exosome, they have actually, you have already put signal for reparation. But when you use Autologus Exosome, you're actually boosting the repair. So this is the beautiful part about it. Okay? Okay, so how do we actually do with Dr. Leroy kit? Okay, so far I'm using Biotech's Exosome kit. See, tak ada gambar ya? Okay, no gambar, sorry. Okay, so dalam Biotech's Exosome kit ni dah ada bottle yang dah ada gel. The gel, it looks exactly sama seperti PRP kit, tapi dia bukan sama. It's not same with PRP kit. Okay, dalam botol ini has a specific gel which Dr. Leroy buat. Gel ini, when you centrifuge in a very high level, gel ini akan mulai pecah. Okay, the centrifugation will be usually at 3,500 to 3,800 RPM. So when you put the tube with the gel that is contained inside of the centrifugation machine, at 3,500 to 3,800 RPM, the gel will mulai separate, okay, it will clash, okay, it will go out and it will start breaking everything, okay. So Kalau for PRP, dia akan mengeluarkan plasma, platelet, sorry. Tapi kalau exosome, dia akan keluarkan plasma. Kerana dalam exosome bottle kita, you will see that it is only a plasma. There is no at all platelet. Okay, all the platelet, all the chemical toxin, everything will drop down. So when you draw the patient blood, we also have created a system whereby when you draw the patient blood, the system is such way that it is sterile. So we'll be giving you the vacuum blood needle, okay? And we will ensure that, okay, in our tube, there's a certain types of pressure. So the way you actually collect the blood and put into the tube also matters, because otherwise, when you do the centrifugation process, the tube may burst. Okay, so there's a pressure that Dr. Leroy created to ensure that the blood is pulled out in a very harmonious way. It doesn't break, okay, it doesn't clot, and it's enough. And yeah, so you get it inside, and then after that you incubate it. You incubate it for 39 degrees Celsius, and usually only 30 to 40 minutes. You don't incubate it higher. The incubation is not together with the kit. The incubation, you have to buy your own. Where to buy them, I will show you. And you can even buy incubator that can cause cold and can also cause hot bath. Okay, so this incubation period is the most important time. Okay, the plastic that we are using is a very high level, high grade 2 plastic that doesn't leach into the system. I mean, into the whole system. Okay, so when you incubate it at 39 degrees Celsius, that is our body respond to fever. So what will happen is it will affect me. It will it will mimic fever effect. It will reduce regenerative exosome. Okay, it will increase the heat shock proteins and anti-inflammatory mediators if you have already preconditioned the body properly, yeah. Okay, and that's when we use that's how we separate the plasma. So at the end, in the tube, right, what you'll be seeing is the end product. So enggak usah lagi seperti PRP di mana anda harus memilih plasma rich platelet. Di tube ini, if contohnya you take in 10 ml of darah, you manage to get 10 ml of darah, dalam satu tube, you will be able to accumulate at least 3 to 4 ml of exosome in one tube. Okay? Okay, so why we do incubation? Okay, it mimics physiological fever condition. Okay, so 39 degrees Celsius is a mild hypothermia. Okay, mild heat stress activates heat shock proteins in platelet, leukocyte, and other blood cells. So if you understand right now, autologous exosome mainly used by blood cells. Okay, HSP function is as a chaperone, is stabilized heat shock protein, yeah. Function is as a chaperone, okay. It will stabilize the protein, dia akan prevent misfolding again, okay, and improve cellular stress signaling. Okay, exosome harvested selepas simple stress ini is enriched in protective and regenerative cargo. Why we do this is sometimes if your body is still inflammatory, then this is the way to actually cancel it out, but it's not 100% cancelling out. Preconditioning is the main 100% cancelling out, ya. Dia akan penuh dengan protective and regenerative cargo. Okay. So mainly it will be HSP 70, HSP 90, and anti-inflammatory microRNA. Again, Dr. Janira is going to repeat what is actually happening here. So sometimes our body, even after preconditioning, may have a little bit of inflammation, or definitely will have inflammation. We cannot run from it. That's how we are aging. Tapi if you have done preconditioning properly, then this is the step to ensure, this is a step that Dr. Leroy created to ensure that even that is actually protected. Okay, so it will enhance exosome release, heat stress trigger exocytosis pathway, including multivascular body. Okay, so tadi kan Dr. Janira bilang how the endocytosis berlaku kan dalam cell. That is actually happening right now during the incubation period in the tube inside of the incubator. Okay, and this transient stress increases exosome yield. So dia akan mengeluarkan lebih banyak exosome ke dalam plasma. improve selective cargo loading. Under mild hyperthermia, cells selectively package more stress adaptive signal, okay? Heat shock protein, antioxidant enzyme repairs RNA into exosome. This makes them more effective in counteracting oxidative and inflammatory environments. Okay? So dalam the exosome kit also we have something called filtration. Kenapa kita ingin filter? So dalam exosome kit tu, when you open the box, you will find 8 tubes. You'll find the needles. You will find 2 filters. Okay, so filter size ni I cannot tell you what it is because that is biotech's what you call that pattern. Okay, it was done. It is actually mainly to make sure that we have only 30 to 150 nanometer size range exosome in the body, in the plasma. Okay, it will remove cellular debris, apoptotic bodies, and larger vesicles that can cause inflammation. Okay, it will ensure a cleaner exosome population, reduce the chance of introducing pro-inflammatory byproducts into the patient. So, when we break everything and we incubate it and then we break it through centrifugation, usually you may have pyrogenic bodies that can cause fever. So this is that's why this filtration system was also introduced to make sure that we remove these pyogenic bodies. Okay, so the first filtration remove large debris. So there's 2 filters in here, guys. It remove large debris, which is the cell fragments, which is the fibrin, apoptotic bodies, microvesicles above than 200 nanometers. Okay, the second filtration has a very fine pore. Okay, below 0.2 2 micrometer or smaller. Okay, this one, as I said earlier, to remove pyrogens, bacteria, and residual aggregates. So when you use this exosome back into our body, there's no any side effect, even if you use it through IV. So PRP, you cannot use it for IV, but this one, yes, you can use for IV. Okay, so for local injection, joint, skin, intranasal, small amount of large vesicles are less risky. Okay, but for IV infusion, any contaminant, microclots, or large vesicle could circulate systematically, raising dia akan buat immune activation, fever, and vascular occlusion clotting lah ya. So that's why IV always require the highest purity standard. Again, when it comes to this tube is not PRP ya guys. This tube will only create exosome. Okay, so we have actually tested from one of our patient. Okay, so if you see there, their bodies that is actually as smaller than 100 nanometer. So that is exosome is from 30 micro to 100 micrometer, right? 130 micrometer, sorry. Okay, so kalau systemic and local routes ialah what can you use for the exosome that we have from this autologous exosome. You can give for intravenous. Intravenous can be used for systemic regeneration because it uses multiple pathway. VEGF pathway is one of it. Immune modulation, anti-inflammatory support. Intra-articular uses the paracrine pathway, which is joint injection for osteoarthritis, cartilage regeneration, ligament repair. Okay, subdermal mesotherapy and intradermal that also using the paracrine, signaling. But again, what I'm going to tell you is exosome is actually very, very intelligent. So even though if you inject in the face, it can also still signal the other pathways. So it can even travel. So if you're thinking that you're only giving for face, be very, very careful. If you don't precondition the body, that inflammation, that signal from the face will move towards other part of the body and signal something wrong. Okay. Intranasal, you can use, it can bypass the blood-brain barrier for stroke, okay, for cognitive decline, Parkinson, Alzheimer's, and many other patients, okay. This is all the special routes. This is practically done by specialists. So if you can do intratical via lumbar puncture. Intramuscular, usually you use ultrasound probe for local tissue injury, myopathy, muscle recovery, okay. Again, you can actually combine many other factors you can put in together to make a topical and transdermal cream. Okay, Again, eyes, ocular, you have to do it under a slit lamps, okay, to look into where you want to actually use it. Mainly macular degeneration is one of the, or retinal degeneration is one of the best thing for you to use. Inhalation, you can use through nebulization, okay, and currently they are plant derived and milk derived exosome for gut. Tapi it's so far like what we are understanding is not going to be effective because that is plant signal into human signal. So okay. Already tell you this is just a recap. Autologous versus allogenic exosome. Autologous, patient own blood. Okay, something like PRP but not PRP. Allogenic is donor, cord placenta, bone marrow grown in lab. Variability high depends on age, health, inflammation, and preconditioning. In allogenic, no, it's a low because it's standardized. It follows the donor cells and it's in a culture condition. Processing, we have centrifugation, filtration, incubation. Okay, where else that is done in the lab. Quantification, again, if anybody tell you autologous can actually be quantified, dear friends, it can only be quantified if you actually precondition the patient and it depends on the patient blood. Example, if the patient is having clotting, like I said earlier, the D dimer is high or the fibrinogen factors is actually not seen, then most of the time you won't be actually getting a proper yield of product. And again, centrifugation also matters, how you centrifugate it, at which level and how long. At the same time, how you also incubate it, all of that matters. And that is all ralat law. That means it's not something that you control. It changes time and again, it changes by who is actually handling, who is actually doing. So hence, we would like to actually come to Indonesia to show you how to use the kit personally, and or we can also do a video online or something like that. On the 23rd and 24th in Malaysia, we are having a masterclass on exosome this month in September. If you come, we'll actually show you how to do it. But we only have limitation of 16 seats. So far, 11 or 12 seats is already taken. So remaining only 4 more seats. Okay. And clinical use is personalized, okay, immunologically safe, tapi potensinya is variable. Again, depends on the doctor, how good the doctor is. Okay, and limitation cannot standardize exact particle counts. So again, this is a review. Patient pre-draw prep, you do the sample collection, you do the incubation. Okay, the incubation, make sure is 30 to 40 minutes and the temperature is only 39 degrees. The duration cannot be more than that. And how you're going to use it for clinical usage. So the main question is going to be here is, everybody's going to ask is like, how long can you actually store this exosome? So maximum hours you can store this exosome if you put it in the fridge, which is around 2 degrees Celsius to 4 degrees Celsius, then you can actually save it for around 6 hours to 12 hours. But if in case more than that, no, you can't. Okay, so why am I saying this? Some people are using exosome for hair, for hair loss. Okay, and majority of the time, if you are going to actually look for the bulk of the hair and you're going to inject it after opening, it's practically something like a hair implant. It will take at least 4 to 6 hours to achieve. So for that patient, yes, I recommend you prepare the exosome. Okay, you prepare all the syringe and you keep in the fridge and you take out as you use. Okay, but if in case that you're using it for face, then prepare the patient immediately, prepare the blood, centrifugate it, incubate it, centrifuge it, and then immediately use in within 1 hour. In within 1 hour, that is the best result you can get. Okay. So we thank you from the bottom of our hearts. Okay, this was actually previously my old slides. I didn't get to change. Okay, again, regeneration begins not in the vial but in the terrain that it comes from. Again, it also determined by the doctors, which is you. Okay, and if you have any more information or for training and booking, you can always look for me or Dr. Vidya. Peace out. Any questions?
Thank you, Dr. Janira.